Today we're going to look at the synapses of chromosomes and meiosis. First, we're going to look at this example of chromosome number 1, where there's a crossover between the genes J and K, and there's a genetic mutation that switches gene ML to the other side and HG to the other side of the chromosome. When the crossover happens, we will have two chromosomes that would look like the parent chromosomes, which will be this upper one here and this orange one here. So we can see they look exactly like their parent chromosomes, and these will be viable. For the rest of them, where the crossover happens, since they're arranged in this manner to optimize exchange of genetic material, the chromosomes will come out differently. If we follow yellow chromosome starting here, we see that we obtain M, L, I, J, K, and then the crossover happens and we move backwards again, getting L and M once again. For the blue chromosome, we start over here, we get G, H, K, crossover, J, I, H, and G once again. Since we have repeated alleles in these two chromosomes, they become non-viable. Now, looking at a second example here, we see these chromosomes look a little different, but chromosomes can be arranged in whichever manner. So if we flip it, it's exactly the same chromosome. Now, if the crossover happens between J and K again, we will have chromosomes that looks like the parent chromosomes, which would be these are the outer ends of a chromosome. And the two middle ones will cross over, but if we follow them, we get J, G, H, K, J, I, L, and M. And if we follow the green one, we get the same thing but backwards. M, L, I, J, crossover K, H, and G. So all of these chromosomes are viable since there was no mutation in this chromosome. Now, the fittest chromosome or the fittest organism in this case would be number 2, since it has the most viable offsprings.